Hi everyone, my name is Elizabeth Ketchmark, and I'm going to be talking to you today about Ninhydrin. So, Ninhydrin is a chemical compound, also known as a triketohydranidine hydrate. Its chemical formula is C9H6O4. I have the IUPAC name displayed for you to check out as well. As you can see by the image on the screen, it consists of one five-membered ring that is fused to a benzene ring. Additionally, it features two ketone and two hydroxyl groups. The two reactive ketone groups are crucial for ninhydrin's ability to detect amino acids. So ninhydrin comes in a solid form that ranges from white to a pale yellow. As it becomes anhydrous or dehydrated by heating, its color reddens. It is most commonly distributed in a crystal or powder fashion. It is soluble in water, meaning it dissolves and disperses throughout the solvent easily. Ninhydrin's molecular weight is approximately 178.14 grams per mole, and it has a melting point of 250 degrees Celsius. Ninhydrin is a chemical that reacts with amino acids, most notably primary and secondary amines, peptides, proteins, and ammonia. It is best known for its reactions with amino acids to create a distinct purple color known as Ruhmann's purple. This is a deep blue/purple dye that forms as a result of the chemical reaction between ninhydrin and amino groups. This chemical is a strong oxidizing agent, causing amino acids to go through oxidative deamination. It is sensitive to light, so it should be stored in a dry, opaque container like an amber glass bottle. Due to ninhydrin's properties and abilities, it serves as a fantastic compound for use in forensic science, biochemistry, histology, and even in the food industry. It can be used to detect, identify, and analyze chemicals, biological, and pathologic materials. Ninhydrin may also be used as an indicator since it changes color. It may also be used as a reagent to detect specific substances like amino acids, as previously discussed. Ninhydrin is most widely known for its use in forensics. Primarily, it is used to develop fingerprints on porous surfaces. Fingerprints leave behind components like sweat and skin residue, which contain amino acids. By visualizing a fingerprint, an otherwise invisible clue can now be analyzed. To develop a fingerprint, an anhydrous solution is sprayed onto a porous surface like paper. The paper is then allowed to dry to prevent running. Think of ink writing, running, and disappearing when you spill water on a document. This is done to prevent that. Once it has air dried, the paper is then heated to accelerate the reaction between anhydrin and amino acids present in the fingerprint residues. The result is a bright purple print that can be photographed and further analyzed to identify an individual. Ninhydrin also serves a purpose in biochemistry laboratories. An ninhydrin test is useful for detecting the presence of amino acids, amines, and proteins in unknown samples like urine, saliva, and semen. This test can help to indicate abnormal levels or unusual traces of such, and is often used in screenings for metabolic disorders. With the use of chromatography, the color intensity can reveal specific patterns that can suggest an issue. In the environment, ninhydrin is not expected to significantly accumulate, especially when in the presence of organic material like amino acids. It is expected to undergo biodegradation in soil. Studies show that the concentration of ninhydrin increases initially in grave soils, but decreased within 1 to 3 years as decomposition occurs. This suggests that this chemical is biodegradable and pH dependent, or it can be processed with the help of microorganisms or by the soil environment. Although ninhydrin is water soluble, it should be contained and not disposed of into bodies of water in order to prevent contamination. Disposal should be in containers through an approved waste disposal plant in accordance with local and national regulations. Ninhydrin's effects on ecology are not super clear. It is not classified as a hazard to the environment, but it could be harmful to aquatic life due to its common solvent mixtures like alcohol. Other resources say ninhydrin is classified as toxic to aquatic life with long-lasting effects and is suspected of causing genetic defects and disrupting crucial biological functions because of its reactivity with amino groups. Data regarding its mobility in soil is also not known. As stated before, it isn't expected to accumulate in soil, therefore it shouldn't accumulate significantly in organisms. Ninhydrin does not have a major effect on the atmosphere, but its reactions release carbon dioxide and ammonia, and its solvent carriers, like acetone or ethanol, are volatile and vaporize quickly. These can contribute to indoor air pollution, so use of ninhydrin requires adequate ventilation. Burning and heating ninhydrin releases hazardous products like carbon monoxide, carbon dioxide, and nitrogen oxides. These are toxic and corrosive fumes that can have severe consequences. Ninhydrin can also be combustible if ignited. Due to these hazards, ninhydrin should never be ignited, and all sources of ignition should be avoided. Ninhydrin is an irritant that is capable of adverse effects if not used properly. It can be harmful if swallowed, causing nausea, vomiting, and diarrhea. It is harmful if inhaled, causing coughing and shortness of breath. Ninhydrin can irritate your skin and cause skin rashes and discoloration. If it gets in your eyes, it may cause pain, redness, or even burns. There is no information available on its carcinogenic effects and if this contributes to cancer or not. There are numerous techniques to measure ninhydrin. Qualitative analysis, like performing colorimetric tests, can detect amino acids and proteins and is observable by a color change. This confirms or disproves the presence and concentration of these compounds. Quantitative tests, like using spectrophotometry and preparing a standard curve, can measure concentration of amino acids. For example, taking protein solutions of unknown concentrations and reacting them with anhydride can be measured for optical density with a blank. Since concentration is directly proportional to the intensity of the color or absorbance, this allows quantification with a standard curve to determine the amount of amino acids in a sample. Now, for the current application portion of this presentation, I found a scientific report called Measurement of Postprandial Peptides in Blood Using Ninhydrin. The main problem addressed in the study is it is unclear what digestion products appear in the bloodstream after eating protein and what products get absorbed. Knowing what components are present could be useful in determining the importance of protein end products in health and disease. Traditional methods to measure amino acids and some peptides, like HPLC, generally are expensive, demanding, miss small peptides, and are inconsistent. The goal of this study was to develop an inexpensive, simple, and reliable method to quantify amino acids and peptides that appear in blood plasma after eating. The analytical process began with a sampling strategy. Postprandial blood samples were taken from pigs, who are a common model for human digestion due to having similar gastrointestinal physiology. Blood was collected before feeding to retrieve a baseline, and after feeding in 30-minute intervals to create a time course curve. These samples were passed through a centrifugal ultrafiltration device to filter out free amino acids, dipeptides, and tripeptides, and to retain proteins and large peptides. Each filtrate was then divided into two analytical subsamples. Sample A, the non-hydrolyzed native sample that would measure free aminos and free peptides, and sample B, the hydrolyzed with HCl sample that contained broken down peptides and measured total amine groups from amino acids and peptides. Both subsamples were then reacted with ninhydrin to produce a purple chromophore. They were then measured spectrophotometrically at 570 nanometers. The analytical technique used was a Ninhydrin colorimetric assay. This is a classic method for amino group detection. The key innovation in this study was the mathematical algorithm that was developed. The algorithm would calculate the free amino acid, dipeptide, and tripeptide components, represented by W in the equation below. By using expected amine group ratios for free amino acids, dipeptides, and tripeptides, we can estimate the relative contributions of them. This turns a chemical assay into a quantitative peptide profiling tool. The main findings of this study found that most absorbed protein products are di and tripeptides, not free amino acids as originally thought. The Ninhydrin and algorithm data showed that longer peptides, having chains longer than 3 amino acids, are not detected in significant amounts. Peak absorption occurred at typical intervals, about 1 to 2 hours after feeding, which is consistent with digestion kinetics. Overall, the algorithm in the Ninhydrin method was shown to be an effective, low-cost, and reproducible tool for nutrition and metabolism research. In the future, this could influence the design of protein supplements, clinical nutrition, and metabolic disorders research. Here are my cited resources, followed by my cited resources for the images that I used in this presentation. And that's all that I have for you. Thank you for joining me today.